Hi, I'm Debra, the CBO of Luminary Therapeutics. We're an early-stage non-viral autologous cell therapy company focused on cancer as well as autoimmune disorders. A few of our accomplishments since formation, which was a little over a year ago. We formed this company with a seasoned team of cell therapy management and scientific team. We raised about $2 million to date and are seeking non-dilutive funding with grants. We are building our pipeline, licensing in proprietary and novel next generation assets with very strong intellectual property. Our first two licenses are for a target for our first product, I'll talk about a lot in a bit, from Case Western Reserve University for hematologic malignancies and autoimmune disorders. We've also licensed in a transposon technology, which is next generation over the first generation from Bio-Techne. And we are advancing our first product towards GMP manufacturing and IND-enabling studies for our first phase 1 study. A little bit more about our unique approach in autologous cell therapy. Our choice of using the transposon is to eliminate some of the obvious issues with viral-based cell therapy, cost, time, getting in the queue, all of those things, but it also ensures that we do not have to test for residual unbound virus in our patient product. The second generation transposons have been shown in other clinical studies to be safer than first generation transposons. And transposons allow for a larger cargo capacity in advanced cell therapy constructs. In licensing, these next generation advances help us to build a very strong pipeline for future clinical studies as well as commercial products. And our early investors are investors that came with many of our founders who founded successful cell therapy company previously and had a successful exit for those investors. A little bit about our proprietary pipeline. So our first product is LMY920. This is a CAR-T BAFF, which allows us to target B-cell malignancies as well as autoimmune disorders. So our first foray into the clinic, and as you can see, we're advanced in preclinical work already, is mantle cell lymphoma, followed by an autoimmune disorder to be disclosed. LMY232 is our universal receptor product, which also allows us to access tumors or cancer, specifically solid tumors and autoimmune disorders. So let's take a deeper dive into BAFF, our first product, a CAR-T-directed BAFF product. This product is licensed, as I mentioned, from Case Western Reserve University, with the ability to take it into, from an IP standpoint, cancer and autoimmune disorders. It is a second generation treatment for B-cell malignancies, of which we know the first generation is CD19 directed CAR. But those CARs eliminate most of a patient's B cells, if not all. We preserve pro and pre B cells, which we think is an advantage from a safety standpoint for these patients. So if you look at the schematic, you can see our engineered T cell with a second generation CAR backbone and our BAFF target. Now, it's important at this point to say our BAFF is not an scFv, it is a ligand, which gives us the opportunity to bind to 3 BAFF receptors, BCMA, BAFF-R, and TACI. Now, there are products that just target these individually. We feel this is an advantage, especially from an antigen escape standpoint. So if we look at traditional scFv targeted CARs, one of the common challenges is antigen escape. You're not directing the product at another antigen except for the one that's very specific. And once the cancer cell loses that antigen, you have no ability to target those cancer cells. With a ligand, it offers us lower immunogenicity potential and with more stability and potentially decreasing the chance of aggregation. With scFv CARs, we know you have to have a linker. And it's been published that those linkers can cause safety issues. Now the question comes up, but you're targeting 3 targets, doesn't that come with it additional safety issues? We believe not, based on our early animal data, but also in the literature, you can see there are other products, ligand-based products, that target more than 3 receptors. So looking at a ligand-based CAR that's directed at 8 targets and at 4 targets in the clinic, so far, no major toxicities or dose-limiting toxicities have been seen. Let's talk a bit more about antigen escape and cell selectivity. So I'm going to use my cursor here. So if you follow my cursor, you can see CD19 eliminates all of these B cells in a patient's system. If we look at BAFF-R, BCMA, and TACI, we preserve the pro and pre-B cells that CD19 takes out, and we just target these later stage B cells. We feel that's an advantage and it's selective. If you look over here, here's a little affinity chart. So here's our BAFF ligand, and each of the receptors and their affinity to BAFF. This BAFF ligand offers us multiple choices in cancer treatment because we're preserving those pre and pro B cells. However, our first foray will be in mantle cell lymphoma, which interestingly is a solid tumor. So we'll show you some early efficacy data in mouse models that was generated by Case Western University that came to us as we're working with the PI there. And she studied ALL and mantle cell lymphoma in her mouse models. So as you can see in ALL, this is CD19 positive cell tumors or tumor cells over here to the left, and the placebo control product, the tumors just grow and found in bone marrow, spleen, and blood. Looking at the mice that were dosed with the BAFF CAR-T in the same tissues, tremendous reduction, almost to nothing in those tissues of the mice. In mantle cell, we gave the mice a mantle cell tumor, and at day 14, we treated them. So at day 13, you can see they're all growing tumor cells nicely. And the blue line is the absolute control. The tumor just grows, grows, grows. The gray line is the control T cell. Eliminates a little bit of the tumor. However, when we give those mice BAFF CAR-T, we've got almost in full elimination of the tumor. Are we the only ones working in BAFF-targeted products? No. We're in good company. Lilly and Novartis are working on monoclonals targeting BAFF, specifically for autoimmune disease, and they're quite advanced in the clinic. But as we know, monoclonals are chronic treatment and not curative. The City of Hope has been working on a BAFF-R, not all 3 receptors, because they're a scFv, so they're only targeted to 1 receptor, and they're BAFF-R. They're in preclinical animal models, so far as it has been published, in B-cell malignancies, and I believe that their hope is to be curative just as we are with our CAR-T. So our differentiation from City of Hope is we're a ligand, they're an scFv. We think that's a significant efficacy advantage without bringing any added safety issues. And for us, we will focus on B-cell malignancies as well as autoimmune disorders. So a sneak peek at our phase 1 study for LMY920 in our first safety study. This will be a basket lymphoma study. Mantle cell, as you may know, is a rare tumor, so would take us quite some time to recruit. So we'll do lymphomas as a basket, all patients who have failed standard of care, standard 3x3 oncology design. We have a design of 4 dose cohorts, and as you can see, going up a log or half a log. Patients, and we haven't reviewed this with the FDA yet, but our assumption is we'll be able to dose 12 to 30 patients given feedback from the FDA. Our primary endpoint, of course, is safety, and secondary is tumor regression or stable disease, and biomarker reduction. We'll be looking at cytokines as well. Our confirmed study sites are Case Western Reserve University, one of the leading hematologic malignancy specialists there, and Case has affiliated cancer centers as well that will participate out of Cleveland, Ohio. So I've mentioned before, we are very focused on strong intellectual property in second generation cell therapy assets. So our first target, BAF, is an exclusive license from Case Western. The patent expires in 2039. So the other thing that's very important to us is longevity of the patent. And this patent includes use in cancer and autoimmune disorders. The transposon is a non-exclusive license from Bio-Techne, formerly Bimogen. And we have the ability to use this transposon in all our cell therapy constructs moving forward. The universal receptor product, LMY232, was internally developed and wholly owned intellectual property by Luminary Therapeutics. The expiry is 2040, also giving us the opportunity to work in cancer and autoimmune disorders. So I've mentioned our experienced scientific and management team. Jeff Leiter is the former founder and CEO of Bimogen and Progenitor Cell Therapy, where he built the company successfully and had a successful exit for his investors. Bo Weber, Brandon Moriarty, and David Legasparta, our three other founders, were founders with Jeff in those other companies. Myself, I have 5+ years in cell therapy and 30 years drug development experience in the industry. And Arik Dudek, who is our chief medical officer, is a practicing oncologist at the University of Minnesota. So, and has lots of experience in cell therapy studies, as well as, of course, oncology, hematologic malignancies, and solid tumors. Just a little summary of our highlights. Again, we're an early stage autologous non-viral cell therapy company with the ability to go into cancer and autoimmune disorders with our pipeline. Our focus is on very novel next generation cell therapy assets, and also with an eye of products that can reduce toxicities over current cell therapies, either on the market or in the clinic. And always to mention, again, our scientific and management team are highly experienced in drug development in the cell therapy space, as well as research. And as most small companies, we're seeking funds to advance our first product into the clinic. Thank you so much for your attention.